Hello and welcome back to the Biochemistry for Health Sciences channel. We'll continue this series, Understand Not Memorize Metabolism, and in today's video we shall talk about glycolysis. In the previous video we explained why this conversion of glucose through this pathway to ATP, so this glucose to ATP pathway, which we call Main Street. We explained why this is probably the most important pathway or highway in your cells. So this Main Street is made up of 4 parts: glycolysis, PDH, the Krebs cycle, and the electron transport chain coupled to oxidative phosphorylation. And in today's video we shall look at the first part called glycolysis. So let us look at the most important features of glycolysis that will not only help you understand the big picture of human metabolism, but also shows how glycolysis plays a very important role in human physiology. And as mentioned before, we'll always come back to the most important thing that human cells need, without which the cells cannot live and therefore humans cannot live, and that is energy. So when we talk about energy, we usually are referring to this nucleotide ATP. As we discussed in the previous video, glycolysis provides from one glucose you get 2 ATPs here and 2 ATPs here, so that's 4 ATPs, but you use up 1 here and 1 here, so use up 2, so 4 minus 2 will give you 2, so that will give you 2 ATPs per glucose. And these ATPs here that are generated in reactions 7 and 10, these ATPs are generated by a process that we call substrate level phosphorylation. And substrate level phosphorylation can happen, ATP can be generated, and we do not need oxygen. So that's one of the biggest strengths of glycolysis. One glucose can give you a net of 2 ATPs without requiring any oxygen. Now, if oxygen is available and the cell has mitochondria, then glycolysis, which also produces NADH. This NADH, as explained before, can enter the mitochondria and provide us with extra ATP. So if the cell uses the MAS system, the shuttle, to transport the cytoplasmic NADH into the mitochondria, then we can get 5 additional ATPs per glucose. And if the cell uses the GPS shuttle, then we can get 3 ATPs per glucose. So that's probably one of the most important strengths of glycolysis. Provides energy to the cell, provides ATP to the cell, even without oxygen present. So glycolysis is a very important source of energy for red blood cells, because red blood cells do not have mitochondria. And so for red blood cells, generating this ATP from glucose via glycolysis is extremely important for its survival. Now, even if cells had mitochondria but they did not have oxygen, then generation of ATP by glycolysis becomes very, very important to the cell. For example, if this neuron here, its blood supply was blocked because of some sort of a plaque or some sort of a clot, and so its blood supply is blocked, there is no oxygen reaching the cell. So although the cell has mitochondria, there is no oxygen. So this cell will use glycolysis to generate its energy so that it can survive for some time. As we mentioned in the previous video, under these conditions, lactic acid will be produced. The pyruvate will be converted to lactic acid, and this is important because the NADH that is produced here in reaction 6 will be reconverted. So the NADH is reconverted to NAD, and this NAD can be reused here in reaction number 6 because glycolysis requires NAD. Okay, so without NAD, which comes from a vitamin, glycolysis will stop. So this means that cells like your red blood cells, and remember we have about 24 trillion red blood cells. So red blood cells will continuously produce lactic acid. So the blood lactic acid that we see all the time, it will be coming from red blood cells because they will continuously produce lactic acid. Now other cells, such as your neurons and muscles, they will only produce lactic acid if they don't have sufficient oxygen. In that case, they will use glycolysis for ATP generation, and that will lead to lactic acid production. So lactic acid in your blood comes from two major sources, your red blood cells as well as cells that do not have enough oxygen. Point number 3, glycolysis has 10 reactions. So let's look at a reaction, the way we see reactions drawn in metabolism. So for example, here we have an arrow going in this direction. So, and then we have another arrow that's joining this arrow. So this basically means in this reaction, glucose is reacting with ATP. Okay, so glucose and ATP are the substrates for this enzyme here, this kinase enzyme here that we'll talk about in a few minutes. And the products that are formed include glucose 6-phosphate and ADP. Okay, so in this reaction, glucose and ATP are the substrates, ADP and glucose 6-phosphate are the products. So that's how we draw the reaction in metabolism. So when you look at the glycolysis pathway, you will see that reaction number 1, you can see the arrow is only going in this direction. And then reaction number 3, the same thing, arrow only going in this direction. And reaction number 10, the arrow is only going in this direction. These 3 reactions are irreversible. That means they can only go in that direction under cellular conditions. The other 7 reactions are reversible, and you can see these arrows are drawn like this, which means they are reversible reactions. So the other 7 reactions can occur in either direction. So for example, this 1,3-bisphosphoglycerate can be converted to 3-phosphoglycerate, or 3-phosphoglycerate can be converted to 1,3-bisphosphoglycerate, because this is a reversible reaction. So under cellular conditions, this reaction can happen in either direction. Point number 4, glycolysis is a catabolic pathway. Now remember, metabolism, when we use the word metabolism, we are basically looking at 2 aspects. One is synthesis or anabolism, and the other is breakdown or catabolism. Okay, so anabolism and catabolism make up metabolism. Glycolysis is a catabolic pathway. So we see a larger molecule. So in catabolism, we are taking bigger molecules, and then we are breaking them down into 2 or more smaller molecules. So here, for example, you see glucose has 6 carbons. So you can see there's 6 carbons here of glucose. And the end result is we will get 2 of these pyruvate. Each pyruvate has 3 carbons. Each pyruvate has 3 carbons. So a big molecule is being broken down, in this case, into 2 smaller molecules, and therefore we call this is catabolism. So this is a catabolic pathway. Point number 5, you can see there is one reaction in glycolysis that requires NAD. So glycolysis requires NAD. So NAD comes from a vitamin. This vitamin is called niacin, or vitamin B3. Remember, we do not make vitamins. So here we have a reaction in glycolysis where we have to depend on a chemical that we do not make, and that is niacin. And we take that niacin and we convert it into NAD. So here is the structure of NAD. So NAD is required. This is required by this enzyme here. And this is therefore, NAD is therefore called a coenzyme. Okay, an organic chemical, in this case required by a protein enzyme, by an enzyme. This is called a coenzyme. And we'll talk more about more coenzymes in future videos as we cover metabolism. Since we do not make vitamin B3 or niacin, and if we could potentially be deficient in niacin deficiency, we do see this in chronic alcoholics who get their calories from alcohol rather than from nutritional food. And so this niacin deficiency is called pellagra. And these individuals with niacin deficiency, they, of course, glycolysis will be affected because this reaction will not happen. So energy generation will be affected. And we do see conditions in the central nervous system such as dementia, they have skin inflammation, and they have digestive problems such as nausea and vomiting. Now the NAD is this part of this form of the coenzyme NAD. This is the oxidizing agent. NADH is the reducing agent. So since glycolysis requires an oxidizing agent, this glycolysis pathway is oxidative. So there's actually oxidation going on. It's an oxidative pathway. Point number 6, glycolysis produces water. So you can see that in reaction number 9, water is also a product. So we can get water not only from our diet, we can also get water from metabolism. Now all 10 reactions of glycolysis are catalyzed by enzymes. Reactions 1, 3, 7, and 10, so these ones here, 1, 3, 7, and 10, are catalyzed by enzymes that belong to a class called transferases. So transferases, these are enzymes that transfer a group, they transfer some group. This group could be a phosphate group, this could be a methyl group, it could be some other group. So basically these enzymes catalyze the transfer of a group from one substrate to another substrate. So if you look at reaction 1 here, you can see that a phosphate is being transferred from ATP to position number 6 on the glucose. So here is the phosphate that came from ATP to give us glucose 6-phosphate. So this is a transfer of a phosphate from ATP to glucose, from one substrate to another substrate. So the type of transferase that transfers phosphate group from one substrate to another, we give it a special name, we call it a kinase. So whenever a phosphate group is the group that's being transferred, then that transferase is called a kinase. So in case of glycolysis, in all these 4 reactions, 1, 3, 7, and 10, a phosphate group is being transferred, and that's why the enzyme ends in a kinase. So hexokinase for reaction 1, reaction 3 is phosphofructokinase, reaction 7, phosphoglycerate kinase, and reaction 10, pyruvate kinase. So in reaction 1, we see the phosphate is being transferred to this substrate from ATP substrate to glucose. In reaction 3, we see the phosphate being transferred from ATP to carbon number 1 of fructose 6-phosphate. In reaction number 7, the enzyme is named in the reverse direction. So basically the phosphate from ATP here is being transferred to this carboxyl to give us ADP and 1,3-bisphosphoglycerate. The same thing is true for reaction 10, where the enzyme kinase is named in the reverse direction. So once again we see a phosphate that is being transferred to this middle carbon to give you ADP and this phosphoenol pyruvate. So 4 of the 10 reactions in glycolysis use the enzyme class that we call transferases, and more specifically kinases, okay, the transfer of phosphate group. So this is a very important class of enzyme, and not surprisingly, because this is sort of related to energy, because we are talking about ATP, and ATP has phosphate groups, has a phosphate group. So transfer of phosphate group is related to energy transfer. 3 of the reactions of glycolysis use the class of enzymes that we call isomerase. And these are reactions 2, reactions 5, and reaction 8. Now isomerases are enzymes that catalyze rearrangement of atoms in the substrate molecule. A common isomerization reaction that's catalyzed by isomerase is the interconversion of an aldose to a ketose or a ketose to an aldose. And remember from your carbohydrate chemistry, aldoses are sugars that have aldehyde groups. Ketose is a sugar that has a ketone group. So converting an aldehyde to ketone is a very common reaction in metabolism, and it's an isomerization reaction that uses the enzyme isomerase. So you can see, for example, in reaction number 2, a glucose 6-phosphate, is converted to a fructose, which is fructose 6-phosphate. Remember, glucose is an aldose, fructose is a ketose. Glucose has an aldehyde group, which you cannot see because of this cyclic form. Fructose has a ketone group, which again you cannot see because of the cyclic form. Reaction 5, you can clearly see the ketone in dihydroxyacetone phosphate, and you can see the aldehyde group which is present in glycerolide 3-phosphate. So the conversion of ketone to an aldehyde, or the reverse direction, aldehyde to ketone. So this reaction number 5 is catalyzed by an isomerase, and to be more specific, this is called a triose phosphate isomerase. So you remember from your carbohydrate chemistry, a triose means a sugar that has 3 carbons. So dihydroxyacetone phosphate has 3 carbons, and so does glycerolide 3-phosphate has 3 carbons. That's why we call this a triose. In reaction 8, in this reaction here, we see a phosphate group going from carbon number 3 to carbon number 2. So basically there's a rearrangement of atoms in the substrate. And this enzyme is also an isomerase. So mutase is an example of an isomerase. So this enzyme catalyzing reaction 8 is called phosphoglycerate mutase. There are 2 reactions in glycolysis, number 4 and number 9, that use this class of enzymes called lyases. So aldolase is an example of a lyase, enolase is an example of a lyase. So lyases are enzymes that catalyze the breaking of bonds, such as a carbon-carbon bond or the carbon-oxygen and so on, without any oxidation or without any hydrolysis. So for example, in reaction number 4, the enzyme aldolase, So in this reaction number 4, this enzyme aldase breaks the bond between this carbon and this carbon. So right here, to give you a 3-carbon unit, dihydroxyacetone phosphate, and another 3-carbon unit, glyceraldehyde 3-phosphate. So this is the reaction where the 6-carbon molecule fructose 1,6-bis-phosphate is broken down into one of this dihydroxyacetone phosphate, 3-carbon, and another 3-carbon glyceraldehyde 3-phosphate. Okay, so this is a reaction where we have the carbon-carbon bond breaking, and that's what makes this a catabolic. You're breaking bigger molecules into smaller molecules. In reaction number 9, that is catalyzed by an enzyme enolase, so if we look at reaction number 9, here we're breaking the bond between carbon and oxygen. Okay, so we're breaking a carbon-oxygen bond, and we are eliminating water. So we are eliminating water and we form a double bond in the product. So this enzyme is called enolase, which is also part of the lyase family. One enzyme in glycolysis is an oxidoreductase, and this is the enzyme glyceraldehyde 3-phosphate dehydrogenase that catalyzes reaction number 6 right down here. As the name clearly suggests, oxidoreductase are enzymes that catalyze oxidation reduction reactions or redox reactions, which involve the transfer of oxygen, hydrogen, or electrons from one substrate to another substrate. So many enzymes belong to this family, oxidoreductase. Examples are dehydrogenases, oxidases, reductase, peroxidase, oxygenase. These are all examples of enzymes that belong to this family called oxidoreductase. Now, this is an important hint. Whenever you see a metabolic pathway and you see these coenzymes such as NAD, which is what we see here in reaction number 6, or you see NADP or FAD, then you can safely say that the enzyme that's catalyzing this type of reaction is an oxidoreductase. Okay, in this case, the enzyme is called glyceraldehyde 3-phosphate dehydrogenase. Okay, point number 11, another important aspect of glycolysis, and that's the connection of glycolysis with this enzyme called lactate dehydrogenase, or LDH. So this reaction catalyzed by LDH, where pyruvate can be converted to lactate or the other way around, lactate can be converted to pyruvate. This enzyme is not part of glycolysis, so this reaction here is not part of glycolysis. It's a separate reaction, but it's closely associated with glycolysis. So why is this LDH reaction important? What is the importance of the LDH reaction? So in case of cells that are generating ATP only with glycolysis, such as red blood cells, we must make pyruvate, the cell must make pyruvate to lactate using LDH, because by doing that, the NADH is converted into NAD, and this NAD can be used in reaction number 6. So as we mentioned, NAD comes from a vitamin, and the cell wants to make sure that it never runs out of NAD in order to generate ATP from glycolysis. Now, another very important function of the LDH enzyme is something we'll talk about in the future. There is a metabolic pathway where pyruvate, and this happens in the liver and kidney cells, where pyruvate can be made into glucose. This pathway is called gluconeogenesis. And in this case, we can take lactic acid, lactate, okay, so the liver and the kidney can take lactate, make it into pyruvate, and then convert this pyruvate into glucose. And this glucose can be sent out into the bloodstream to sustain the energy generation that is required by red blood cells as well as brain cells. So the brain and the red blood cells rely very heavily on glucose. So making glucose by gluconeogenesis is a way to make sure that the brain and the red blood cells have enough ATP, enough energy production, especially during starvation states. So these are the 11 points that are important about glycolysis. There is one more very important aspect of glycolysis, and that is regulation of glycolysis. But we shall leave this for a future video because I think this will be best appreciated once you understand the whole of metabolism and you are able to see the big picture. So we'll leave regulation of glycolysis for the future. So since this video series is called Understand Not Memorize Glycolysis, one of the best ways to appreciate glycolysis is by looking at glycolytic enzyme deficiencies. Okay, so this will be it's a subject that we'll talk about in the next video. And when we look at the glycolytic enzyme deficiencies, you will really appreciate the importance of glycolysis as far as human physiology is concerned, as far as human survival is concerned. Okay, so until the next video, goodbye, and once again, have a very happy and blessed day.